Hi everyone, I'm Javeria. I'm a PhD student at University of Science and Technology, UST, and I'm working as a graduate student at CURAMS. We are developing combination radioimmunotherapy for solid tumor. Here we use the combination drug losartan to enhance trastuzumab delivery and to figure out the therapeutic efficacy of copper-64 trastuzumab in gastric cancer mice model. So radioimmunotherapy is a combination of monoclonal antibody and radioisotope. It exploits the monoclonal antibody to target to specific antigen type and thus deliver the cytotoxic radiation to the tumor. We know that cancer cells express particular antigen on their cell surface. In case of hematological tumor like that of B-cell lymphoma, CD20 is overly expressed. Similarly, solid tumor can express particular antigen on their cell surface. For example, in case of gastric cancer and breast cancer, HER2 antigen is overly expressed. So radioimmunotherapy for hematological tumor like that of lymphoma and leukemia is commercialized and is successfully used in clinics. So here are few FDA products that are approved for clinical use. Zevalin, which is a combination of atrium-90 and ibrutinib and can target CD20 antigen on non-Hodgkin lymphoma. Bexar can also target non-Hodgkin lymphoma CD20 antigen and is a combination of iodine 131 and tocilizumab. However, there is no product for solid tumor nature. This is because radioimmunotherapy delivery to target site in solid tumor is limited. It depends on various factors which limits the therapeutic efficacy of radioimmunotherapy. These factors include cancer-derived factor, which comprise of tumor microenvironment, radiation-dependent factor, alpha-beta radiation, bystander effect, or molecular size binding affinity of the antibody. Altogether, it limits the pharmacokinetic and dynamic parameter of radioimmunotherapy. So here I will discuss about tumor microenvironment of gastric cancer, which is the fifth most frequent cause of cancer. Tumor microenvironment is a repertoire of normal cell that is recruited by cancer cell for its growth, progression, and to evade an immune response. The component of tumor microenvironment consists of cancer cells that are tightly packed together, abnormal vasculature, infiltrated immune cells, and extracellular matrix that consist of collagen and hyaluronic acid. Here we are focusing on a drug that can modulate the extracellular matrix for better drug delivery. So the paper published in Journal of Cancer in 2017 shows elevated collagen content in gastric cancer as measured by picrosidase red stain and collagen 1 staining. We can see the elevated level of red stain that depicts collagen content in gastric cancer is increased as compared to the normal tissue. So in our study, we used various methods. Firstly, we prepared a xenograft mice model. It consists of NCI and H7 cell line. It is gastric cancer cell line in a cell concentration of 5 into 10 power 6 cells per 100 microliter. We injected this cell line solution subcutaneously into the nude mice. So for penetration study, we used Alexa 647 fluorescent dye and conjugate it to the trastuzumab. We injected this conjugated solution to the tail vein with or without losartan. After 5 days of losartan treatment, the tumor tissue were excised and is further processed for microscopic imaging. We also measured interstitial fluid pressure using experimental apparatus which consists of a transducer probe with the needle. The needle was firstly injected directly into the tumor tissue and was retracted, making the probe static in the tissue to record the reading. So for collagen content, we used empty stain and polysaccharide content of ECM was stained by Elsin blue stain. Further, we used immunofluorescence and primer-specific QRT-PCR. We further carried out radioimmunotherapy comprising of copper-64 trastuzumab with or without losartan to acquire PET-CT scan imaging consecutively for 5 days. PET scanner uses copper-64 emitted positron for imaging. The generated PET data was reconstructed for OSEM 2D reconstructed algorithm. We have also performed assays for apoptosis like 3D cultured cell viability using Alarmer Blue assay, PI stain, and flow cytometry. So MMP assay was carried out to measure total matrix metalloproteinase using protein sample for tumor tissue. Western blot for SERPINE1 antibody and quant-seq analysis were also performed from tumor tissue. Trastuzumab can bind to HER2 antigen on gastric cancer tissue. We showed here penetration of trastuzumab as indicated by green signal both from the vessel and from the edge. With the addition of losartan, the green signal is increased from the vessel and from the edge. We showed 1.2-fold increase of trastuzumab penetration from the vessel and 2-fold increase of trastuzumab penetration from the edge. Also, the total accumulation per tumor area was significantly increased in case of trastuzumab plus losartan. This result shows that with the addition of losartan, monoclonal antibody, that is trastuzumab, can reach out to further HER2 antigen across tumor tissue. So our result shows improved RIT uptake in combination treatment of copper-64 trastuzumab with losartan. The image here shows standard uptake values from copper-64 trastuzumab and copper-64 trastuzumab with losartan. The combination group of RIT with losartan shows increased signal. Also, the SUV max for combination treatment with losartan was increased at 96 hours and 120 hours significantly. So our result shows that losartan reduces ECM component in gastric cancer tumor model. This figure here shows empty stain for collagen. The collagen is stained in blue, and we can observe that control groups show higher collagen content as compared to the losartan treated sample. Similarly, ECM polysaccharides as stained by addition blue stain and is shown in blue color was observed to be decreased in losartan treated sample. So these results of decreased ECM component by losartan could explain the increased trastuzumab penetration and copper-64 trastuzumab delivery across gastric tumor tissue. So in this figure, we found that with losartan treated sample, the interstitial fluid pressure was reduced. Since in tumor microenvironment, the cells are tightly packed together with elevated ECM components, which generates high interstitial fluid pressure. This pressure serves as a barrier in drug transport. By reducing the ECM components, the pressure is relieved and drug transport can be increased. So our result shows that losartan can induce cell cytotoxic effect and cell cycle arrest. As we observed in 3D culture cell viability assay, increased propedium iodide stain by losartan, and we also observed increased accumulation of cell concentration at G0 G1 phase. So this explains the increased trastuzumab penetration by losartan and enhanced copper-64 trastuzumab in combination treatment because of the potential of losartan to remodel the extracellular matrix, induce cell cytotoxic effect, and dropping down the interstitial fluid pressure. So we evaluated therapeutic efficacy of combination treatment by carrying out tumor volume and survival study with groups like PPS, trastuzumab, losartan, losartan plus trastuzumab, copper-64 trastuzumab, copper-64 trastuzumab plus losartan. We find significant reduction in tumor volume and improved survival rate in case of combination treatment of copper-64 trastuzumab with losartan. This result shows that losartan possesses therapeutic efficacy in gastric cancer. So to explore the underlying mechanism, we carried out quant-seq analysis at 1-day and 7-day loss-treated tumor sample. We find that SERPINE1 was significantly downregulated by losartan treatment. Our gene network analysis shows the network connection of SERPINE1 gene with TGF-beta1 and ECM components like collagen and MMPs. Therefore, we suspect the signaling mechanism of losartan-regulated ECM degradation via TGF-beta1 signaling. So in this figure, we quantify mRNA level of TGF-beta1, SERPINE1, MMP2, and 25. We find that losartan treatment significantly downregulated SERPINE1 and TGF-beta1, whereas it upregulates MMP2 and 25. So the downregulation of SERPINE1 might increase MMP activity. MMP plays an important role in ECM degradation. So we confirm the SERPINE1 downregulation by losartan in 3 tumor tissue at antigen level. The image here shows immunofluorescence stain for SERPINE1 colored in green. The green signal for losartan tumor tissue was significantly reduced Also, the Western blot data for protein sample we observed reduced band in case of losartan treated sample. We also confirmed significant upregulated total MMP at protein level by losartan treated sample. So to summarize, we showed here that losartan promotes deeper and more uniform distribution of copper-64 trastuzumab in gastric cancer and thus limit the radiation damage to the normal tissue. Gastric cancer is overly expressed in ECM network, therefore it masks the HER2 antigen. Losartan reduced the collagen and ECM component and possibly exposed the hidden HER2 antigen for copper-64 trastuzumab targeting. Losartan is already approved as a safer clinical drug and thus is an ideal candidate for combination radioimmunotherapy in gastric cancer. So in conclusion, we showed that losartan reduced ECM, improved trastuzumab delivery, and potentiate copper-64 trastuzumab in gastric cancer. Thank you so much. That's all. I'm done with my presentation.